Yellow. We're talking neuroscience today. The word of the week is neurobabble. Google it. So, in the last lecture, I set up this premise that every major movement within the history of cognitive psychology was a reaction to what came before it. So, behaviorism was a reaction to structuralism's focus on these sort of abstract, vague, subjective concepts of elemental sensations and using this very questionable technique of analytic introspection. And we swing completely in the other direction towards, studying things that are unobservable, directly not observable, to studying behavior, which is the only thing that we can see and measure. Cognition was a reaction to behaviorism because behaviorism is like everything inside the black box is irrelevant. Cognition, we can't deny that there are mental events. And so that's the reaction. And I made the argument right at the end of the last lecture that um, cognitive neuroscience is actually a reaction against cognition. Cognition was the sort of really looking at it from this very box and arrow model of mental events was very popular from, I want to say, like the 1960s. Neisser's book published 1967 until into the 90s, right? When cognitive neuroscience, when neuroimaging started to become widely available and used for these research purposes. And I actually want to say I'm projecting into the future here, and I do think that I see a reaction against this. So cognitive neuroscience was incredibly popular because it was attractive to, I think, a lay audience. The idea of people talking about brains, there's something very technical or authoritative about the idea of bringing brains into the discussion. But I do think that there is a reaction against cognitive neuroscience. We'll talk at the end of this lecture about where that might be coming from. But the reaction that I am now seeing against cognitive neuroscience, like I think cognitive neuroscience was the thing from late 90s through to maybe like the 2010s. I started to see a softening of the celebration of neuroscience probably in the 2010s. And now I do think that I'm starting to see this reaction against the idea of like, we don't necessarily need pictures of brains in order to say something about cognition. We don't need to necessarily talk about where in the brain something is happening in order to understand cognition. But what I do think is happening is I'm seeing a reaction against the idea that cognition is something that happens exclusively in here, whether you talk about it as the mind or whether you talk about it as the brain. I think the next reaction is coming in the form of embodied cognition. The idea that cognition happens not just in this black box, in this brain meat structure, it happens in our entire body. And so embodied cognition is the theory that many features of cognition are shaped by aspects of an organism's entire body. And so, for example, I have a very specific example, which is why we're looking at this picture of an escalator. One of the elements of embodied cognition, or one observation from embodied cognition, is that it's harder to walk up an escalator that's not moving compared to a stationary set of stairs. And the reason is because our body has that expectation that the escalator is going to give us that boost. And when we don't get that boost, it actually makes us feel like we're working even harder. So whenever I see an escalator that's not moving and like a set of stationary stairs, I take the stationary set of stairs. I know that's going to be easier because my brain isn't making the same assumptions about what's going to happen in my body. And what what's another good one? Distances seem further if you're carrying something heavy. So if you're asking people to make a judgment about distance, that number varies based on whether or not they're holding something heavy, or if their hands are free. So there's all sorts of fun observations that come out of embodied cognition. But I'm just stepping out on a limb. I don't know this is actually the next iteration or the next reaction, but this is my intuition. On top of that, I'm adding, like, I think a global societal anxiety around artificial intelligence and what that means for us. And I think that one of the fundamental differences between artificial intelligence and human cognition is that we have bodies. So the idea that we might be able to, like, store our consciousness on a computer server after we die in order for our consciousness to live on, I don't think it would be us. Cognition happens in our bodies as much as it happens in our minds. Okay, so today's agenda, we're going to be talking about mental representations, neuroimaging, functional anatomy, localized versus distributed representation, which is part of functional anatomy, but is a specific point that I really want to make. And then at the very end of class, we'll talk about what neuroscience tells us about cognition. So I wanted to start with this revisiting of the SOR framework. The O, what is that O? You see something, that's the input, that's the S, then what? The answer, according to cognition, is mental operations. Information is transformed, manipulated, mental operations are happening. The brain fires in response to a stimuli. So let's talk about how these mental representations are formed in the brain. I want to start with this question. What are the symbolic representations of the number 4? If you were in a foreign country where you didn't speak the language and you wanted to communicate to somebody that you needed 4 of something, what are all of the different ways that you might be able to do that? So 4, literally you could just, like, do 4. You could write down 4 lines. You could write down the Roman numeral 4. You could write down the alphanumeric 4. You could do, you don't even have to do this, right? Some places it's gonna be this. Anyway, there's all sorts of ways that we can represent the number 4. Literally, the word for FOUR is a symbolic representation. There's nothing like, there's no fourness about that word. These are all symbolic representations. They stand for something else. There's nothing inherent about the alphanumeric that represents fourness. We've all just decided that this alphanumeric means 4, and we've also all decided what 4 equals, what that means also. Even the word itself is a symbolic representation. Okay, so the mind is a system that creates representations of the world so that we can act within it. When we are talking about cognition, we're never dealing with the direct stimulus, right? If I take this glass of water, mentally, this glass of water is not in my brain. There's a mental representation of the glass of water in my brain, and that's the thing that I manipulate. That's the thing that I act on in the world. I'm going to just show this short video of my apartment to illustrate a point of this idea of the principle of representation. I want you to think of your eye, your fixation, as being where that central bullseye is. And, like, if you were looking around a room, if your eyes were darting around, that's the information that is coming in through your eyes. But that's not your experience of the world, is it? You're not constantly feeling like your world is shifting. That's amazing. That is genuinely amazing. The fact that what is the raw sensory input coming in through your eyes is not what you experience is amazing. So while this is what your eye movements are seeing. This is the actual representation that you experience, right? Like your world is stable. So this is the principle of representation. What we experience as perception is not a direct reflection of the external world, but rather a mental representation of it. And this, interestingly, I think is an incredibly detailed version of the world. This is probably not at all what we're representing. This is more likely. So even though it feels like you have a full and rich representation of the world, in reality, if I stopped you, forced you to close your eyes, and then asked you details about what you were just looking at, like these are the two things that you could really tell me about. So the principle of representation is not just that this very frenetic input gets turned into something rather stable, but it's also this richness that we think we experience is actually not really there. If you probe deeply, you would see that there's a lot of this information that we're just not processing or representing at all of these mental representations are the result of activity in the nervous system. And this module reviews how the brain represents specific aspects of our experience. How does activity in the nervous system represent our world? Everything a person experiences is based not on direct contact with a stimulus, but representations in a person's nervous system. One question cognitive neuroscience is trying to answer is how does the brain create these representations? And we're starting with the premise that the activity, it's in the activity of neurons in the brain. So one of the things that you're going to see in cognition is representations of that activity as an action potential. We're really going to stay at this very high level representation of action potentials. In intro psych, you probably went through the movement of ions across the membrane that generates the action potential in neurons. We don't really need to do that for cognition. That level of detail is actually not super relevant. It's not going to come up again in the course. I'm going to keep it actually at this really high level of representation where you can see at the bottom of the figure, there is that full representation, that graphical representation of the action potential, the depolarization, the repolarization, and the hyperpolarization. All of that whole process, which by the way is like 1/1000th of a second, 1 millisecond, all of that takes. Still blows my mind. But because we're not really focused too much on the ions, we're going to represent all of that entire process as a line on a timeline. So like a single vertical line is representing an action potential. We're going to really stay at the level of patterns of activation. So the question that cognitive neuroscience is really trying to answer is what's the organization of a brain that allows action potentials to equal me, your room that you're in, your view? How do neurons firing in this meat sack equal our experience? It's a really good question. One method that researchers have used to try to find out the answer is single cell recordings. So during single cell recordings, you take animals, it's a surgery, so you're going to have them anesthetized. And while they're asleep, you're going to do some microsurgery. You take a little cork out of the skull and insert an electrode into the brain until you encounter the body of a neuron where you're literally measuring directly from that neuron. And the idea here is that the microelectrode then can do this continuous measure of activity, and you wake the animal up. Still, it's a delicate procedure, so they're going to have stereotaxis. Their head's going to be held still, but while you're measuring from that individual neuron, you're then showing the animal different things in order to see what stimulus actually provokes a response in that particular neuron. And so you can see A is representing probably a baseline firing rate. So there's spontaneous firing rates of neurons across the brain. It means that increase in action potentials, firing rates, or a decrease in firing rates are both informative. So A is probably that spontaneous level. B suggests that the researchers are now showing the animal something that that neuron is particularly interested in, that it's specialized for processing. So you see the activity level increase. C, just getting even a little bit stronger in the overall reaction time, or the overall reaction of the neuron. So using this technique, researchers tried to figure out the organization of the visual cortex for vision. And they were literally measuring neural responses from neurons in the primary visual cortex using cats as their model. So I've got these two side-by-side cortices here. The left is the cat, the right is the human. And I'm showing you where the visual cortex is in the cat. The primary visual cortex in humans is in the same place. Now, I've artificially made these similar in sizes. Our brains are much bigger than cat brains, but it does seem like there's a lot of overlap or similarity between the way vision works in humans and vision works in cats. So we can use them as an animal model to try to understand more about how vision works. So measuring neuronal responses from neurons in the primary visual cortical of cats, researchers found that each neuron in the visual area of the cortex responded to a specific type of stimulation presented to a small area of the retina. So first of all, when you're measuring from neurons in the primary visual cortex, each neuron sort of cares about a different area of space. So you have to put it in a particular location. But then also on top of that, each neuron cares about a particular area of space and a particular kind of feature. So researchers found that using this technique, so measuring from the primary visual cortex of cats and then exposing the cat to a variety of visual stimuli, they found that there were areas of the primary visual cortex where neurons had a preference for a particular area on the retina, but also within that area, there needed to be presented a bar of light of a particular orientation. Other neurons preferred a bar of light of a particular orientation and moving in a particular direction. Some preferred a bar of light in a particular orientation, moving in a particular direction and of a particular length, so maybe short rather than long. And the researchers who discovered these neurons called them feature detectors. So feature detectors respond to specific stimulus features like orientation, movement, and length. That was something of a surprise. Originally, researchers in this field were actually exposing the cats to very complex stimuli. Like they were exposing them to like shapes and circles and all sorts of different stimuli, and they couldn't get the neurons to fire until one of the researchers accidentally left the slide projector on as he was changing slides, and it was the line on the slide as it was being dropped into the projector that made the neuron fire. And suddenly they realized that they had just gone too far, that they were trying to get the neuron to fire with full stimuli, when in fact at the primary visual cortex, those neurons really just prefer basic features, not shapes, not even objects, but like parts of shapes that would then make up objects. Here, I'm going to actually illustrate some of the more contemporary or more recent findings using this technique of single cell recordings and moving into primates, so using monkeys as opposed to cats. Researchers were also looking for areas of specialization in the temporal cortex to try to figure out, like, what does the temporal lobe do? And I'm showing you these stimuli at the top 1 through 6. The number actually indicates the relative level of firing for a particular neuron in that area of the temporal lobe in the monkeys. So 1 produced very little firing, these basic shapes of circles, squares, rectangles. But once we start to get some points on the stimuli, we now see that neurons in this part of the brain are starting to fire a little bit more strongly. And then as we move towards hand-like stimuli, we now start to see these neurons really firing. A human hand produces a very high level of response, but a primate monkey hand produces the strongest level of response in these neurons. What this suggests to us and to researchers in general is that the primary visual cortex processes basic features, but as you move forward in the brain, those features get sort of put together in order to create more complex stimuli representations. Here's another set of results from single cell recordings, again using primates and the monkey temporal lobe. There's an area in the temporal lobe that is specialized for processing faces, and using these stimuli that are illustrated on the left, whether it was human or monkey faces, you would see massive activation in this area. In comparison, when you showed it non-face stimuli like places like a skyline or a bush or a building, you would see actually neurons in that area of the brain fire less and potentially even be suppressed a little bit, showing us that we're seeing specialization of neurons, and now that specialization is just more complex. It's not based on simple features, it is based on now significantly more complex objects or categories. Okay, so with this idea of neural representation, what we've seen at a model level, so the theory or the model of what we think is happening in the brain is something akin to what is happening in cognition, where we have features that are being joined together into a conjunction of features. So a bar of light gets conjoined with another bar of light to produce a corner. A corner gets joined up with other corners to compose a shape. Multiple shapes get conjoined together in order to create an object. As you move forward in the cognitive processing, you get this hierarchical organization where you start out with very simple features and eventually get up to full objects. We see a parallel process happening in the brain. In the primary visual cortex, you've got features that are just being processed. So a vertical edge or a horizontal edge is being processed in the primary visual cortex. You move forward a little bit and those feed into a neuron that's processing corners. You feed forward a little bit more and that's feeding into a neuron that now can represent a full rectangular surface. You add that together and eventually you get to a set of neurons in the primary or in the temporal lobe that can recognize a full object of a table. This approach, this representation that we're looking at right now actually seems to imply that there is something called a grandmother cell, right? That all of these neurons will converge so that they're at the very highest level. There's going to be a single neuron that specializes in processing a table, but that's actually inaccurate. That is not necessarily what this is meant to communicate. What this is meant to imply is hierarchical organization in the brain. However, what we see at the level of the cortex is not specificity coding. It's not one neuron equals one concept. in and this would be the example of specificity coding or the grandmother cell theory. With this approach to neural coding or this theory of neural coding, the idea is that every single concept is represented by an individual neuron. So in the brain, we have a series of neurons that we use to represent different objects, and each object is a particular neuron. Bill is represented by the firing of neuron 4. Mary is represented by the firing of neuron 9, and Raphael is represented by the firing of neuron 6. Specificity coding proposes that a particular object is represented by the firing of a particular neuron. But obviously, this would be disastrous. It is not a very robust system. If you have a weekend of hard partying and you drink a little too much and you kill off neuron 4, suddenly Bill doesn't exist. This is essentially what was being assumed with symbol manipulation models from that conference at Dartmouth. But it also seemed to sort of be implied by that graphic that we were just looking at, but this is not what's happening in the brain, and it's not actually anything that anybody thought was really happening in the brain. Specificity coding in the grandmother cell theory was proposed as a comparator to what we actually think is happening. So another possibility for coding, or how it is that these neuronal firing come to represent different kinds or categories of objects, is distributed coding or population coding. That's another term that people use for this. So with distributed coding, you have these neurons in a system, but all of the neurons are becoming active, and it's the pattern of activation across the system that actually comes to represent the different concepts or individuals. For example, Bill would be one pattern, Mary another, and Raphael another. Imagine here you lose neuron 4. Kind of doesn't matter. You still have the overall pattern of activation. So this system is much more robust. It's robust against damage. If you lose neurons, probably the pattern is still preserved. The other advantage that you see with population or distributed coding is that a small number of neurons can actually come to represent an infinite number of stimuli. So it's very economical. This is essentially where the conversation ended when I was in undergrad. I make it sound like I'm so old, and honestly, I'm getting up there. But this is true. We talked about specificity and distributed coding and left it at distributed coding. But the story has progressed since then, and in fact, where we've kind of landed is somewhere in the middle. I find that's usually the case when we have this dichotomy, is eventually the discipline settles on some sort of happy medium. And that happy medium is called sparse coding. So with sparse coding, you still have specificity. So you have localization, you have these areas of specialization, but you're still seeing a pattern of activation. It's just a pattern of activation amongst a much smaller area of maybe specialized neurons in the brain. So sparse coding helps to explain areas of specialization in the brain without resorting to the extreme of grandmother cells. Let's move on to neuroimaging. When it comes to neuroimaging, there are so many different techniques these days. There are somewhere between 10 and 15 different techniques, but my experience is that they fall into two broad categories. In cognition, because there's not, honestly, because I'm actually really kind of focusing on a very behavioral cognitive approach to the course, we're not going to actually look at a ton of research that uses very specialized techniques. So I'm really keeping us at this core, we're kind of staying within the core level of neuroimaging and these four major techniques. But where I do think it's worth investing some time is in this distinction between structural neuroimaging and functional neuroimaging, because those 10 to 15 techniques, and even the new ones that are being developed right now, they're in all likelihood going to land in one of these two categories of structure versus function. So structural neuroimaging, I have this icon here of a photograph or an image, because structural neuroimaging is literally about looking at the structures of the brain. Does everything look like it's there? Are all the physical pieces present? And we're going to look at CT and MRI. These are, two of the most popular methods of structural neuroimaging. On the other side, we have functional neuroimaging. So functional neuroimaging is actually giving us a view about what's happening in the brain. I have a film strip here because it's not just a static image. It's actually, dynamic processes, what's happening. And the two major techniques that can tend to get used a lot in the field is PET, positron emission tomography, that traces radioactive glucose, and then fMRI, which tracks deoxygenated blood. In both cases, researchers are essentially looking at where food for thought goes. PET it's tracing where glucose goes in particular, so where sugar is being consumed. fMRI is tracking where oxygen is being consumed. In both cases, we're making the assumption that wherever resources are being consumed in the brain is where the work is being done. One of the very first neuroimaging studies, structural and functional neuroimaging studies that was published in the literature was Phelps and Mazziota in 1985. They had a patient who showed evidence of cognitive decline. And after his death, so they did both neuroimaging before he died, and then after his death, they were capable of doing a postmortem. So in the postmortem, there was no evidence um, of damage. So everything looked pretty normal. Using a CT scan, they were actually able to find evidence of structural damage subcortically, but they didn't find any damage to the cortex. However, while the patient was still alive, positron emission tomography PET scanning demonstrated widespread abnormalities of the frontal cortex. They found that activity was reduced by 21% and on the left hemisphere, the left frontal lobe, compared to the other side of the brain, compared to the right side. And I mentioned earlier that PET is tracing glucose consumption. The way that we do this is using a radioactive dye, essentially. So glucose is attached to a radioactive molecule, and then that tracer is inserted into the system. It circulates in the system, and what's being traced is as that radioactive molecule decays, it emits positrons that are then used to build an image of the brain of where glucose is being consumed in the brain. And so you can see that when it comes to PET, positron emission tomography, you actually get a fairly degraded image. It's not a super clear anatomy image, but you can see where activity is being being performed. And if you're waiting for positrons to be released from this radioactive tracer, you can imagine that that takes some time. So you have to wait for a PET image to be built up over a few seconds. And so even though it can give you an idea of function, it has to be function over the course of a few minutes, seconds, minutes at least. And so it has to be something that's sort of continuous. You can't really trace out, for example, where I don't know, like a very short perceptual process might be happening using PET because it doesn't have great temporal resolution. And you know what, PET doesn't even have very great spatial resolution these days compared to functional activity overlaid on an MRI image. That's kind of what usually is people are doing today. They'll take an MRI and then they'll do the PET and they'll overlay it so that it just like looks a bit clearer. And this is where we start talking about MRI and fMRI. MRI is, first of all, it's the machine, and an MRI machine has this giant magnet in it. At the top image, you can see a person that you lay down and you're inserted into this large metal tube. Once the metal tube is, electricity is applied to it, it creates this incredibly strong magnetic field. So that whole thing is just a giant magnet. And what happens is that the atoms in your body are usually all randomly oriented, but when you turn the MRI machine on, this giant magnet aligns all of the atoms in parallel with the electromagnetic field that's being generated. And then the MRI tech sends a radio wave to ping the atoms and knock them over. Then what gets measured is the rate at which they realign. And different density of tissue is actually going to realign at a different rate, which allows you to create that image. MRI, in comparison to CT and PET, you're getting these beautiful, really clear structural images. And it's costly though, and it's prohibitive for a lot of people. It's a giant magnet, right? So um, people with any sort of metal implants can't get them. Soldiers, if you've been in an area of active war, they generally won't give you a MRI unless they do a full body X-ray first, because they want to make sure that there's no like shrapnel or just random pieces of metal in your body, because this will pull them out. There's some dental work, especially older dental work, that like people won't be allowed to be in an MRI machine because it's just such a powerful magnet. And there's so many safety standards around MRIs because it's a giant magnet. But it gives you these really beautiful, clear images. You can see on that bottom figure, there's that area of brightness, and that's just showing us that there's probably, I'm guessing that there is a stroke or a bleed there, and so that tissue just appears brighter because of that lesion that is forming there. So fMRI, it's most similar to PET. It's because it's looking at where resources are being consumed in the brain, but it's actually specifically measuring oxygen atoms and where the most oxygen is being consumed in the brain. Now, an fMRI, sometimes people call it a type of MRI, but that's not really true. An MRI is a static image, whereas an fMRI is looking at function, and they're using this machine in different ways. So yes, they both use an MRI machine, but an fMRI is actually taking advantage of the fact that hemoglobin, which is a ferrous or an iron containing molecule, carries oxygen in the brain and is sensitive to electromagnetic forces, right? Because it literally has iron within it. And so fMRI is tracking that hemoglobin molecule, making the assumption that hemoglobin is carrying oxygen to areas of the brain where it's needed most. And so it's looking at where blood is being deoxygenated. Deoxygenated blood just behaves differently compared to oxygenated blood. So it tells us where oxygen is being consumed. This image here I'm seeing, first of all, often fMRI images use color coding to indicate like level strength of response. And so I'm seeing the strongest levels of response in the occipital lobe, and then some levels, some responding in the temporal lobe and towards the medial part of the temporal lobe. I'm guessing that this is an image from a study looking at object recognition. So primary visual cortex is being stimulated, but you're also getting activity in the temporal lobe, which is particularly interested in object identity. So fMRI has better spatial and temporal resolution than PET. But yeah, it's prohibited from some people and very, very expensive. That's one of the main barriers for people to use fMRI in their research. No, it's this way. It's this way. Go do some stuff. Or you could save it all for later. I don't care. All right, let's talk more about basic functional neuroanatomy. We're going to start with this part of the brain, the cortex. This is actually, I think, what people think of when they think of brains. They think mostly of that cortex, that pink, wrinkly part that's right on the top. And what are the purpose of the wrinkles? The wrinkles actually allow more surface area in a smaller volume of space. So brains of other animals like reptiles are almost completely smooth. This is the brain of a monkey, and you can see even in other primates, humans actually have an incredible level of folding that's happening here. So more so than even some of our closest living relatives. And so, why do we have this level of wrinkling? And the answer is that it gives us more surface area. So, the cortex is really only the outer, like 2 to 3 millimeters of the brain. In fact, cortex means bark. So, it's like the bark of a tree. It's very thin, but it's where all the hard stuff is being done. And so, how do we get more of this without growing our brain or growing our skulls to such a size that it would like break women? And the answer is you scrunch it up, right? So if you look at this handkerchief here, it's a lot of surface area. I could not put this in my pocket unfolded, but you fold it all up and suddenly no problem, that's going to fit in your pocket. Same thing is happening here. That folding is just giving us more surface area. So we just have a way bigger napkin compared to other mammals and even other primates. Probably you guys have heard about like left brain versus right brain processing, and I saw this graphic all over social media a few years ago, right? The idea is that the left brain is super sciency and analytical, and the right brain is very like creative and artistic. And there were all sorts of like surveys that you could take. Are you left brain or are you right brain? This is some straight up bullshit. In other classes, I used to say to students, if you write this on an exam, I am going to have a serious talk with you. Because there is specialization to the hemispheres, but this like oversimplification of like the left brain is science and math and the right brain is like creativity and arts, it's just it's oversimplified. It's so oversimplified, it's wrong, and it really bothers me. Also, the tests about are you left brain or right brain, good news everybody, good news, you probably have both. Amazing, right? Yep, most of us have both. All right, so what is true? What is true? That is that, yeah, okay, so the cortex is divided into two hemispheres that are approximate mirrors of each other, but they actually do have different interests, different specializations. The left hemisphere is specialized for processing analytic information or sequential information. So anything where the stimuli needs to be processed in isolation is going to be in the left hemisphere, and anything where sequence matters. So, for example, math is going, order of operations matters, that's going to be in the left hemisphere. Lots of language is going to be processed in the left hemisphere. So, for example, Christy kicked Priya versus Priya kicked Christy, different meanings. The only difference between those sentences is the order of the words. So, sequence here really matters. The right hemisphere, on the other hand, is better at processing holistic information. And so holistic is like big picture and processing multiple pieces at once. And it's also interested in spatial processing. Spatial processing is by definition holistic. You only know that somebody is at the front of a room because there is a back of the room. North Van is only north if you're comparing it to Vancouver downtown. But if you're comparing it to Squamish, it's south. So space is always relative and holistically processed. So the right is going to be processing like navigation, processing maps, for example. And music-related abilities are often processed in the right hemisphere compared to the left hemisphere, although I will say that people with musical expertise probably do show some processing of music in the left hemisphere, because for musicians, sequence matters. For music listeners, we tend not to hear notes, we tend to hear melodies, so we're processing it much more holistically. All right, so the cortical lobes, you've got these two hemispheres. You got a left and a right. They are slightly specialized, but then when you look within each hemisphere, there are four major cortical lobes. So the occipital lobe, the temporal lobe, the parietal lobe, the frontal lobe, and then you can also see in this image the brainstem and the cerebellum. Those are subcortical structures. So the cortical lobes are there for processing sort of higher level conscious function. The occipital lobe is specialized for processing vision and visual kind of stimuli. The way that I like to remember that is like in cartoons. Remember when people get hit on the head, on the back of the head, they'd see stars? That's literally, you might actually see visual stimuli if you got hit on the back of the head because you're activating the visual cortex externally, manually, but yeah, so it might produce visual experience. The temporal lobe sits right beside the temples, and the way that I remember its specialization is because, first of all, it sits right beside the temples, sits right near your ear, and what means two things. What means like, I'm sorry, what did you just say? Because the temporal lobe does sound, auditory processing, but the temporal lobe, what also means like, what is that? Like, what is that thing? And it also does object recognition. Now, I used to think these two things were like unrelated. I was just like, why would the temporal lobe do both auditory processing and object recognition? That's crazy. No, it actually makes total sense because one of the things that auditory processing is particularly concerned with is speech and speech and words. And it turns out that object recognition and categorization language are intimately linked, right? We only know, in some cases, you only know that a chair is a chair because you've labeled it a chair, right? It doesn't look necessarily like a stereotypical chair, but you know it's a chair and you call it a chair, right? So like words and objects actually very intimately linked with each other. So then you've got the frontal lobe, sits right at the front, easy peasy, does all of the hard stuff, planning, organizing of information, memories, decisions, inhibiting automatic behaviors, personalities, kind of like in the frontal lobe. So yeah, all the hard stuff. And then the parietal lobe is just the other one. It's just the other one. It's the one that's hard to remember what it does or why it's there, but parietal lobe does spatial processing in particular, and where your body processing. So if you touch yourself, it's where the somatosensory cortex is, but it also is interested in like where things are on the body and then where things are in space. It's a lot of wheres. Okay, now I want to talk about localization and distributed representations. So this is a very tricky topic, I find, because it feels as if localization and distributed representations are at odds with each other, but they're actually complementary. I grew up in Alberta, and watching local commercials advertising Wetaskiwin's Auto Mile. It's a collection of auto dealerships. You can see them on this map, and that are all very close together, and they advertise together. Cars cost less than Wetaskiwin. Literally, that jingle is burned into the brains of all Albertans, I think. And so why would this collection of auto dealerships, who are not part of the same company, they don't sell the same kinds of cars, why would they all collaborate and in order to create this auto mile? And also, what does this have to do with cognitive neuroscience? And the answer is localization of function. Localization facilitates specialization. The reason why these auto dealers are collaborating is because they're actually not competing with each other, or they're all selling very specific cars that there's no overlap. And what it means is that each of these dealerships can get very specialized in what they offer. And so people will come to this particular location because they know they're going to be able to find what they're looking for. And it allows each one of these dealerships to get very specialized. They don't have to stock all of the sort of typical cars. They can get a little more diverse in what they have to offer because everybody's covering basically all of the stuff. Okay, so what does this have to do with cognitive neuroscience? And localization of function gives you more specialization. Some of the earliest evidence for localization of function comes from neuropsychology. So, neuropsychology, people often get neuropsychology mixed up with neuroscience, but it's not the same. Neuropsychology is something very specific. It's a branch of psychology that studies the behavior of people, usually with brain damage. It can sometimes be special populations too. So, for example, people with ADHD or people with autism might get studied under neuropsychology as well, but it's basically atypical brains are being studied with the goal of inferring something about how cognition works in the general population. Some of the earliest reports that had traction came from a doctor named Paul. So near the end of the 19th century, clinicians were really starting to discover that the brain was organized with some very specific functions in very specific areas. And these areas specialized in dealing with specific aspects of different stimuli. But this was kind of stunning at the time because philosophers and scientists hadn't yet connected the idea that activities of the mind could be linked to activities in the brain. It took a long time for us to get there, my friends. But Paul Broca was a pioneer here. He described a group of patients who had a very specific set of speech difficulties. So Broca's patients following stroke would have laborious speech. Their speech was semantic but agrammatical, which means like you could kind of understand what they were trying to communicate, but their grammar was really problematic. And we have since gone on to name this speech deficit Broca's aphasia, or it's also referred to as production aphasia. A few years later, another clinician, Carl Wernicke, described a group of patients who also had speech difficulties, but they were a completely different set of speech difficulties compared to Broca's patients. So Wernicke's patients had fluent speech, grammatical, but they were incoherent. And we went on to refer to Wernicke's patients as having Wernicke's aphasia or comprehension aphasia. So Broca's patients, he eventually did the postmortems on patients that died and discovered that they had common damage in an area in the frontal lobe very close to the motor cortex, which makes sense. Broca's was production aphasia, and the motor cortex is where you're producing speech. So having damage close to the motor cortex could in fact impact speech. Also seems to impact grammar. Wernicke's patients, on the other hand, all seem to have damage that was localized to an area of the temporal lobe. The temporal lobe more about listening and hearing, and here we see problems with comprehension.
Now tell me what this thing was with your legs last week or week before.
No good. Ache and knees and ankles.
What'd you do about it?
Home, doctor, and legs. Walking, no good.
No good, huh? And you're a fellow who likes to walk, too.
Oh,
Jeff.
jeez.
Huh?
Oh, boy. Oh.
Laborious language, semantic. You could understand what he was trying to communicate. He's having some pain in his legs. But agrammatical. Laborious, semantic, but agrammatical. Definitely seeing it. That's an example of Broca's patient. Now let's see an example of Wernicke's.
They're afraid, boom.
What's this?
The big end right there. End of the football.
End of the football?
It influenced the football right here. The cause that it comes in, see?
Yeah.
The end of it, see? Yeah. Oh, we got a lot of these, see?
I don't understand.
Well, it's quite pliable together, see? You don't understand it, but it causes in a long tree comes from there. And it comes source from the country out through, see? All the under it, see, it comes up and finally come out here and goes out, see? Yeah.
So with Wernicke's aphasia, fluent speech, he's not struggling to get the words out. Grammatical, his sentences actually have the right structure, but incoherent. Literally almost the diametrical opposite of Broca's patients. So with comprehension aphasia on this side, right? So what we see here is a double dissociation. So we see like these two different groups of patients having diametrically opposite deficits, telling us that these are separate and independent components of language. Grammar and production is in one area, and comprehension comprehension and semantics is in another area. And so this was the first kind of inkling that what was going on here is specialization in the brain. Before that, we literally did not have a really clear idea of the correlation between activity in the brain and activity in the mind. Since then, we've discovered that actually we have a number of modules in the brain. These are areas of the brain specialized for specific functions. And it's usually categories of stimuli that are important for the human experience. And so they're given specific space in the brain for even more specialized processing, because that localization gets us more processing power. And there's three modules that really exemplify this idea. There's the fusiform face area, the parahippocampal place area, and the extrastriate body area. So the fusiform face area, damage to this area of the brain leads to prosopagnosia, an inability to recognize particular faces. Sometimes in the popular media, like journalists will refer to this as face blindness, and this is an image that I found when I was doing an image search for prosopagnosia. But people with prosopagnosia don't walk around seeing skin faces, so this is a bit of a misrepresentation. But this is probably a little bit more accurate. People with prosopagnosia see faces. they just can't tell anybody apart based on faces alone. The features all look very similar. There's generic eyes, there's a generic nose, mouth, etc. And there's actually a couple of really great episodes on Radiolab on prosopagnosia. And so I'm just going to like point you to those episodes because they're so good. One's called Letting Go, and the other one is called Strangers in the Mirror. And they're first person perspectives, and they talk about some of the relationship difficulties that pop up when you have prosopagnosia. But I'll let you guys seek those out. So, looking at the brain, we're going to be looking at these modules where they're located. So the fusiform face area is on the posterior part of the temporal lobe, so it's at the back part of the temporal lobe. And if the brain, like if you think of this as like your hands together as kind of like a brain, if you flipped it over, you could see that the fusiform face area is just on sort of that knuckle of the temporal lobe that sort of folds underneath. It is usually bilateral, but if it's not bilateral, then it's lateralized to the right hemisphere. Holistic processing. And we also can see here the parahippocampal place area. So the parahippocampal place area is located in the medial temporal lobe. It's literally tucked up even further compared to the fusiform face area. But again, an area that's specialized for processing a particular category of objects. And it's actually not even objects. The parahippocampal place area processes places. And just before we move on from this, you can see the extrastriate body area is just outside of the primary visual cortex. It's still within the temporal lobe, but it's in that extrastriate area, which is why it's called the extrastriate body area. And what these stimuli process, you have to go with what the name is. The parahippocampal place area processes places. If you're like, what's a place? Anywhere that you think is a place. Beaches are places. Hotel lobbies are places. Mountains are places. Bedrooms are places. Literally, anything that you think of as a place is going to be processed in the parahippocampal place area. And here, so again, we're seeing that image so you can localize where the parahippocampal place area is. Here in this, this is probably an fMRI image. You're seeing a part of the brain. Imagine like you're slicing it kind of like a loaf of bread. You're seeing sort of that midsection. And on bilaterally, you're getting that activation of the parahippocampal place area. And what the preferred stimuli are, literally anything that's a place. So this picture of the desert, a house, a corner of a room with nothing in it is a place. And then also this Lego setup that kind of mimics a little bit of a skyline. In comparison, objects don't stimulate the parahippocampal place area. The same images, sort of broken up into their constituent parts or sort of pixelated, will not produce the same activation. And then a picture of a Lego that is just sort of abstractly put together, nothing. But if it looks like a place, the parahippocampal place area will become activated. The extrastriate body area, again, not a trick name here. What does this process? Literally parts of bodies. So we're seeing bilaterally that activation in the fMRI image. Again, with one of those like bread slices images. And any illustration of the human body or part of a human body is going to activate the extrastriate body area. But faces don't work. Faces have their own place. And, the same stimuli but rearranged so that they don't look like a body will not stimulate the extrastriate body area. It has to be a recognizable human form and like a body form, not just a face. And even the implication of a body will activate the extrastriate body area. This image of a person. Got more stuff for you. Go do this. Or wait, save it. I don't care. Okay, so then that's localization, right? Specific functions are served by different areas of the brain, and in processing our experiences, often stimuli are going to elicit multiple specific functions. So, for example, first of all, a face is going to activate the primary visual cortex. It has to. You're using your eyes to see it. But then also it's going to activate the fusiform face area, definitely. However, that face is also at a specific location, and the location of objects in the environment is coded in the parietal cortex. If you recognize this particular face as belonging to the actor Paul Walker, that's going to recruit part of the temporal lobe. You might actually feel a bit sad because you realize that he died very young. And that is probably going to be processed in like the frontal cortex and subcortical structures, a lot of subcortical structures. So the idea here is that we actually do have, first of all, areas of specialization. The fusiform face area is specialized for processing faces. That doesn't mean other parts of the brain aren't involved in processing faces. You literally have to see them with your eyes. So there's definitely going to be all parts of the visual cortex that are going to be involved. But the point is that recognizing faces has a very special area in the brain. It's not the only area that processes faces, but it's going to be specialized for processing faces. On top of that, you also have this idea that any cognitive task actually recruits multiple cognitive functions. Sure, face recognition is in a specialized area of the brain, but you don't often just recognize faces in isolation. You recognize faces in certain contexts while you're doing certain things. And because those tasks are recruiting multiple cognitive functions, you're going to see activity all over the brain. So yes, we have localization of function, but we also have distributed representation of stimuli and experiences. So fundamentally, stimulus processing recruits multiple cognitive functions, activating multiple areas of the brain. For example, if you have a person who's watching a red ball rolling across a table, you're going to see activity all distributed all over the brain because there's multiple cognitive functions that are being recruited, and even those cognitive functions are going to be using processing from other areas of the brain to help them, even if with their specialization. So if you want to isolate a particular function, you actually have to do a subtraction to get rid of all of that distributed representation. And the way that you do that is you create a second condition where the only difference is the cognitive function that you're interested in. So if you're interested in tracking where emotion is being processed in the brain, you're going to first present participants with a ball rolling across the table. Then you're going to present exactly the same scenario, but now the ball is stationary. The idea here is that you can now subtract out any of the activation that is not common to both scenarios, and that's going to tell you where processing of motion is in particular. This is the basically distributed representation is why PET and fMRI have to use the subtraction method. So this is a certain cognitive activity. If you're interested in a certain cognitive activity, you have to compare it to a baseline where a person is basically doing something, doing nothing. Sorry. The subtraction technique, if you're interested in a particular target activity, like motion, you want to take the combined activity of everything all together minus that baseline in order to get at that target activity. So if, for instance, you're interested in figuring out where in the brain manipulation is happening, you need to come up with a combined activity. So first of all, you can't manipulate anything without holding it. But then you need to partial out that baseline activity in order to isolate manipulating separately from holding. And so that would be the baseline activity, is literally just holding a stimulus without manipulating it. That will tell you where manipulation is in particular. So this bottom image with the subtraction out, whatever is the leftover activity tells you that that's where manipulation is being processed in the brain. This is very similar, by the way, to Donder's mental chronometry. We talked about this last time, where the idea is that we have two different versions of a task, the only difference being the insertion of a decision. And using the subtraction, it can tell you how long it takes you to make a decision. This is the temporal equivalent of that, and the subtraction technique for PET and fMRI is the spatial version of Donder's mental chronometry. I really strongly recommend having a conversation with generative AI. If you have any questions about this, what's the difference between localization of function and how is it different from distributed representation? Yeah, I just think that's a very useful approach to take if you're feeling confused about a topic. More stuff for you guys to do. Lots of stuff this module. All right, last but not least, what does neuroscience tell us about cognition? This is interesting. So I'm going to start with this question about what is critical thinking. I think people use it all the time. I hear faculty say that they want students to learn critical thinking, but then like, would you be able to recognize it if you saw it? So let's dive in. What does critical thinking actually entail? First of all, being skeptical. Skepticism isn't like, I don't believe you. Skepticism is like, how do you know that? Why do you think so? Informed by evidence. Disciplined thinking is rational, open-minded, and informed by evidence. Where are the receipts at, my friend? Recognizing errors and biases when one's own thinking. So the idea of actually like engaging in reflection on your own mental processes. Do you have errors? What assumptions are you making? Looking for logical relationships between ideas, and then drawing warranted conclusions and generalizations. All right, so we have this basic definition. Let's put it to work. So when I originally taught this course, I did have a textbook, and I was using Goldstein's cognitive psychology. And at the beginning of his cognitive neuroscience chapter, he has this section, Why Study Cognitive Neuroscience? And honestly, it really kind of pissed me off. Right at the top, it says, first paragraph, some students taking their first course in cognitive psychology are surprised to find that studying the mind involves both behavioral experiments and physiological experiments. Why, they ask, is it necessary to learn about the operation of the brain to understand how people perceive or remember? For other students, especially those who have studied the brain in a previous course, it's obvious that taking a physiological approach provides important insights into how the mind works. Is it obvious, Goldstein? Is it? We are going to question these assumptions and biases that we have. And I'm going to actually start with the question, like, is it necessary? Not why is it? Let's start with the question of, is it? Is it really necessary? A lot of resources will talk about levels of analysis, and it refers to this idea that a topic can be studied in a number of different ways, each approach kind of contributing its own dimensions to our understanding. And I will agree with this statement, but does it actually imply levels? And does it imply bidirectional contribution? Cognition can be studied at a behavioral level and at a physiological level, but do these inform each other? Here's the example that Goldstein talks about in the textbook. He talks about driving, and I love this example. He says that the idea of like you can test drive a car. It can tell you a lot about the acceleration, the braking, how it corners, its gas mileage, all of that. But we can get different information by looking under the hood. I agree with that. But here's where the analogy actually, I think, it either breaks down or it does its real job, which is that I do not need to know how my car works in order to drive it. It certainly helps. I've learned some stuff about how cars work since I've owned a car, and it helps that I know how my car works. But at the same time, I drove for like probably 15 years without having any idea how any of it worked. And I was fine. I was a good driver even then. So do we need both levels of analysis? And do they really contribute? So do we really need the physiology? That's the question here. Like, yeah, sure, we can test drive with behavior, but do we have to understand the engine? Do we have to understand the brain to know something about how cognition works? So again, one of the examples is that from Goldstein's textbook is the idea of like Gil walking past a park where he recently met up Lisa, and walking past the park reminds him of Lisa because that's where he last saw her. We can talk about the different levels. We can talk about the neurotransmitters that are involved in the processing. We can talk about which neurons become active. We can talk about systems level brain storage. And then we can talk about like how that storage becomes active to produce the conscious experience of memory. But we knew about memory, we knew about associations well before chemical processes, neurons, brain storage, or system level. We didn't need to talk about the brain in order to talk about associations and the mind and memory. We didn't need neuroscience to tell us that memory exists on a cognitive level. It doesn't tell us how you remember having met somebody. Like, just literally because you can describe this doesn't mean how, it doesn't tell you how it happens. Let's see. The truth of the matter is that despite what Goldstein and other resources have said, controversies about, like, for example, like whether or not episodic and semantic memory are actually separate and independent cognitive functions came up way before cognitive neuroscience was a formal discipline, and cognitive neuroscience hasn't settled that debate. It's questionable whether cognitive neuroscience has settled any cognitive debates. And it's questionable whether cognitive neuroscience has actually changed the way we think about cognitive functions, or whether it's just actually enhanced questions about like brain pictures and brain geography. So I'm being a bit of a devil's advocate here. I have a degree in cognitive neuroscience, and cognitive neuroscience is one of the courses that I can teach. And I do think that cognitive neuroscience has contributed to the rich picture of human cognition. But I want you to question the obviousness of how studying the brain can give us insights into the mind. There are lots of researchers who actually really question what it is that neuroscience has offered to cognition. It seems to be a one-way street. This is an argument that is made most strongly by a researcher named Max Coltheart, and I have like an academic crush on Dr. Coltheart. I think he is an amazing logician. He's very logical, and he makes this argument that like knowing where something happens in the brain doesn't mean you know how it happens. The analogy that I like to use, and it's because I noticed years ago that there was a badminton racket factory in Richmond. Taking the SkyTrain to KPU Richmond, I noticed this BK Knight factory, and I used to play badminton in high school, and I just remember thinking like, oh, that's interesting. I didn't know that they made badminton rackets in Richmond. That's just interesting. So cognitive neuroscience is the equivalent, it seems to be the equivalent of asking, hey, do you know how they make badminton rackets? And getting the answer, yes, they make them in Richmond. That's not how, that's where. And where still doesn't tell you how. Do this thing. All right, so it's the end of this module. You should be able to explain the idea of symbolic mental representations, explain the notion of neural representations and the role of hierarchical organization in complex representations, describe the single cell recording technique and explain how it's used to identify neuronal processing, compare the concept of specificity versus population versus sparse coding, recognize research questions that involve structure versus function, explain the basics and recognize the appropriate application of CT, PET, MRI, and fMRI, summarize the basic organization of the cortex, including the functional location of hemispheres and lobes, distinguish the anatomical and functional differences between Broca's aphasia, Wernicke's aphasia, and recognize novel examples of each, explain hierarchical organization, predict functional deficits that could result from damage to specific cortical modules, explain how localization of function and distributed representations are not mutually exclusive, and describe the concept of levels of analysis, and then critically analyze the claims of cognitive neuroscience.